Work on transporters, excitatory amino acid transporters, and was recruited to the University of Pittsburgh, where she's now a member of the Pittsburgh Center for Pain Research. We had some time to overlap there before I came here. She has an abundance of high impact papers in a variety of areas on looking at transporters and now in the pain field, and has gotten sucked into spinal circuitry of pain. Also has a number of foundation grants supporting her work, both in the pain field and in her interests in Parkinson's disease and otolaryngology, when she's not in the pain field. And she's taken on one of the most challenging areas in pain, in the pain field, which is the organization of the mechanical pain circuitry in the dorsal horn that leads to chronic neuropathic pain and allodynia. And she's going to tell us about her most recent work.
All right. Well, yeah, thank you, Derek. Hello, everyone. Thank you for having me. So yeah, Derek and I, we go back. It was actually pretty. I was really happy when I saw Derek's face when I arrived in Pittsburgh, because I had been in San Francisco for a number of years. And oh, louder. Oh, I had been in San Francisco for a number of years, and so I have fond memories of being in the Vollum Institute. And I think most people do. And Derek was one of the characters, in that scenario. So yeah, and anyway, I appreciate, this opportunity to tell you about what we've been working on in my lab. So as Derek mentioned, my lab actually does work on a number of different things, but now we've kind of, we're sort of focused in on pain and specifically on dorsal horn circuits for mechanical allodynia. And so that's what I'm going to tell you about, is kind of our view of the circuitry for mechanical allodynia from the perspective of the dorsal horn. But, of course, the dorsal horn isn't sitting in isolation. It's an actual area of integration. So now we're becoming interested in the circuitry in terms of primary afferent inputs and descending inputs, and also looking at projection neurons, which I was talking to just when we were coming up here. I'm not going to be talking about the projection neurons today, but just in the little bit that we've been looking, and of course, if you look back in the literature for the last 30 years, it's kind of it's actually a little daunting to see how the area of the spinal cord that we're studying, laminas 3 and 4 primarily, you can find connectivity to almost every brain region. So, usually they make, it's simplified: lamina 1 neurons, lamina 5 neurons, spinothalamic tract. But anyway, I don't think it's definitely not that simple. But it brings me to a point that I should probably make here, which is that most of the circuitry that we're looking at is focused on output neurons, lamina 1 output neurons. So it's the circuitry that leads to the activation of those lamina 1 neurons. So just a framework for what we're trying to do is we're trying to understand how high threshold afferents, go to mainly the upper lamina 1, 2, and this is just, in dogma, and activate these pain projection neurons, while the low threshold afferents innervate in the deeper lamina and mediate, or thought to mediate, aspects of touch. And what we're trying to figure out is, when there's injury, how is it that touch or low threshold afferents can then activate this nociceptive network to cause pain? And so I made this little cartoon to try to give, an idea, which is probably not necessary for you guys since you study pain, but the way that we think about mechanical allodynia in a simplified way is that it's basically that the same concept that you have activation of these high threshold afferents that can activate the lamina 1 neurons, and then the low threshold, oh, sorry, I screwed this. This angle is like kind of odd for me. I hope it's okay if I do this like on a cruise ship thing, because I think it'll be easier. All right, this will be better for me. It can interact with you too. All right, good. All right, so this is the model of like how people in general think in a simplified way of how mechanical allodynia works. So it's basically the idea that you have high threshold afferents. I might have to start this over again. Apologize. We'll get this going. All right. Let's see if this works. Maybe it's this thing's not working. Okay. Maybe I can't do a cruise ship presentation. All right, we'll get this working. Okay, so anyway, it's basically the idea that you have these high threshold afferents and they're activating the lamina 1 neurons and you get pain, and the low threshold afferents can't activate the lamina 1 neurons because of inhibition, whether it's tonic inhibition or it's feed-forward inhibition. They don't have access to these lamina 1 neurons that are pain projection neurons. But then in the setting of injury, there's different mechanisms which cause disinhibition. When the disinhibition happens, now these low threshold afferents have access to the nociceptive pathway and can cause pain. So that's the really simplified framework for that. And so what we're interested in, if we want to understand how this is working, is identifying dorsal horn populations that are involved in mediating the mechanical hypersensitivity or allodynia, primary afferents that are involved in this process. We're also interested in the descending inputs, and as well as the mechanisms of induction. So, here I showed, a couple of interneurons, and then they become inhibited, and that's the disinhibition, the inhibitory interneurons. But, clearly, by the end of my talk, I hope you'll be convinced, and if you look in the literature, you'll be convinced, many people, that there's different mechanisms for the disinhibition that can happen, that can lead to the activation of the circuitry. And so another question is, how does this state maintained? What are the different mechanisms for that? And so I title my talk about the etiology and like searching for the etiology in the spinal cord, because one of the take-home messages that I'd like for you to get or to pay attention to is that, my hypothesis, or my lab's hypothesis, is that different types of injury lead to the activation of the dorsal horn mechanical allodynia circuitry, in different ways. So there's basically, we started with this idea that there's a pathway for mechanical allodynia in the dorsal horn, as I've illustrated by my simplified cartoon. But the way we think about it now is that it's actually a network. So that's where we're going with this talk. But my introduction actually into the pain field came by making a knockout of a vesicular transporter. And when we knocked this transporter out globally in mice, the phenotype that we found that related to pain was this one, which is that, so the mice in an inflammatory model or a spared nerve injury model, they don't develop the mechanical hypersensitivity, or they don't show mechanical hypersensitivity. So this is von Frey thresholds. So we have knockouts, wild types and knockouts, wild type and knockout. And so you see there's reduced hypersensitivity compared to wild type mice. And so we looked at other aspects of somatosensation in these mice, and the only other defect that they had was like a reduced sensation to pinching, so like a acute mechanical pain defect, but everything else was normal. So we didn't find defects in itch or thermal sensation or thermal hypersensitivity. So it's really just limited to mechanical hypersensitivity. So what is a vesicular glutamate transporter? So it's basically a transporter on the vesicle membrane that packages glutamate into synaptic vesicles for regulated release. And there's 3 of these in mammals, V-glutes 1, 2, and 3. And they have, so V-glutes 1 and 2 are doing most of the work, and they have, for the most part, a complementary distribution, so they don't overlap that much. And then V-glut 3, which is the one that I knocked out, is expressed much more sparsely in the nervous system. And so when I left my postdoc, I knew that these global knockouts had a defect in mechanical hypersensitivity, and I wanted to figure out, could we use this, because it's so specific, this defect, and it's also so important, this type of pain, can we use this to try to figure out something about the circuitry for mechanical allodynia? And so to do that, what we needed to do was figure out where this defect was if we were going to learn about the circuitry. So if we found where the defect was, we could start to study, the circuitry. And so what we did, and I'm not showing you all the different crosses that we made, but we basically took a conditional knockout for V-glut 3, and we took advantage of all the great Cree lines that the somatosensory system has made. Like you can get rid of, you can delete protein or genes in primary afferents, or you can delete, in just the spinal cord, or you could delete in both spinal cord and primary afferents, and et cetera, et cetera. And anyway, we took advantage of that, and we did all these crosses. And in the end, it turned out that it was a population of neurons, these neurons here in the dorsal horn, that when we deleted them with this mouse line where it just has this Cree recombinase in the spinal cord, deletes V-glut 3, then it recapitulated, basically the phenotype that we saw in the global knockout mice. And so it pointed to these spinal cord neurons here as being the origin of that defect. But one thing that was unexpected with this is that, I don't know how much you guys do these genetic mouse manipulations, but essentially what we've done here is we've taken a mouse that expresses Cree recombinase from the V-glut 3, driven by V-glut 3 regulatory elements, and then we crossed it to this floxed tomato fluorescent reporter mouse. And when you do that, you're basically from the time of, from the very earliest stages, you're going to recombine to activate this tomato reporter in the neurons that are expressing Cree. So when we're looking at this mouse in the adult, it's possible that these cells are actually expressing the V-glut 3 and therefore the Cree early in development and not in the adult. So we had to go back and look at that, and it turns out that these neurons in the spinal cord, they're excitatory interneurons, and they express this transporter only transiently during development. So the transporter expression comes on around P5, and then it's gone by P15. And so then that left me at a kind of juncture, because I was thinking that, these are excitatory neurons, and most of the neurons in the dorsal horn use V-glut 2. And I'm not going to get into the weeds about V-glutes because, that's kind of like for people that care about V-glutes, but it was just kind of an unusual situation. Normally, when we would see V-glut 3 expressed transiently during development, it was in GABAergic neurons, and it was providing a source of glutamate and plasticity, via the, for the development of an inhibitory, synapse. And here we have what's an excitatory neuron anyway, and for whatever reason requires the expression of V-glut 3 in order to, set up the circuit for mechanical pain, essentially. Since, that's the defect we get, mechanical pain defects. So I could go in the direction of studying V-glut biology and development, or I was told I should go in the direction of studying, the circuitry for mechanical allodynia as it relates to any kind of, knowledge for translation in treating pain, which is actually what I also wanted to do. I just was kind of torn. So we decided to go on and see what what this was implicating, or what this was suggesting, is that these neurons are in the circuit for mechanical allodynia, even though, we're not directly looking at that, but that's what it's implying. And so to address that, we used this excitatory dread. So this is a receptor which was designed by Brian Roth and his group, and it allows for the activation of specific populations of neurons depending on, the neurons that you target it to, basically. And it uses a ligand which is supposed to be innocuous, but, there's been some, if you might be aware of, there's been some controversy about the use of the CNO, because it can be degraded to clozapine, and, that's itself a drug of, that people use in psychiatry. But, you just have to do the controls, and we did the controls, so I think it's fine. But essentially the idea is that this isn't going to activate anything endogenous, and this isn't going to cause any problems, isn't going to be activated by anything that's endogenous. And it's really the coupling of these two things that allows for the activation of the neurons. So we targeted this receptor to these VGLUT3 neurons by injecting the receptor packaged in an AAV virus. So we do intraspinal injection into the dorsal horn in these Cree mice, and we did it at P10 because, like I told you, the expression is transient, and so the Cree expression is also transient. So we inject it, and then we just asked, what happens if we turn on these neurons to mechanical sensitivity. And sorry, I have that here. So basically we give the CNO, and now we get a drop in threshold. So the mice have become hypersensitive just by activating that population of neurons in the spinal cord. And then there was no effect on thermal. But, a better way of testing whether these neurons are in the mechanical allodynia pathway would be to shut them down. We didn't have that at first, but then we got it. And so that's why I show you the second experiment, which is that if we use this inhibitory dread, so it's the same principle, but now we're using, the other one was G protein coupled. This is actually an ion channel. So there's a chloride channel, and it's been specially designed to be activated by this ligand, and this ligand is designed to not activate any other, proteins endogenous. And that's by Scott Sternson at Genelia Research Campus, is the one that designed this. And it's very powerful, and we were really excited to give it a try. And so we similarly packaged it into an AAV, injected intraspinally into the spinal cord of these VGLUT3 Cree mice, and then we get the expression pattern here shown. And then we did some in vitro testing. So why we were excited about this dread is because the other inhibitory dread that's used is another G protein coupled dread from Brian Roth, and that one is not always very powerful to shut down neurons. And I was a little bit concerned that we wouldn't get good inhibition in a if the spinal cord has been injured, if there's been a peripheral injury and now the spinal cord's hyperexcitable, it might even be harder to shut down because it's relying on these second messenger cascades, and that there's ion channels, potassium channels, or whatnot that it can couple to. Anyway, bottom line is, whatever the explanation is, it's just not as effective at shutting down neurons. But this ion channel was exciting for us because when this was published, basically Scott's lab took channel rhodopsin, which is the light-activated ion channel, and this channel, and they put them like as a tandem construct, so not fused, but that you would make them both off the same transcript, and they put that in neurons, and then they shine light, they shine light on the cells, and the cells fire like crazy because of the channel rhodopsin activation, and then they put the ligand on it, while they're doing that, and it just goes quiet. So even with this like robust activation by channel rhodopsin, you could shut down these neurons with this chloride channel. So that seemed more promising. And then we did this is just some of the proof of principle for us, where we're firing the neurons that have the dread, and then when we put the ligand on, they go quiet, and the neurons that aren't expressing the dread are unaffected by the ligand. And so then we, ask the question, what happens when you inhibit these neurons in the different pain models? And what we found is that in an inflammatory model, this is static, a static punctate allodynia. I know I'm going to get in trouble for saying allodynia, but it's a nociceptive reflex. But anyway, this hypersensitivity, there's not really actually a rescue by the ligand, but when we did the dynamic, which is like a cotton swab across the paw, then we get a robust inhibition. And then again, in this nerve injury model, we don't see really a rescue for static or punctate, but we get a robust inhibition for dynamic. So this was a little bit of a surprise to us, but actually, because the global knockout has both dynamic and static are robustly, attenuated. So what this was saying to us, and then it was also corroborated by Chufu Ma's group at Harvard, who published it in Nature Neuroscience this past year, is that this particular population of neurons, when you shut it down, seems to be important for dynamic allodynia, but not really having it's dispensable, one would say, for static allodynia. Why the knockout had a static phenotype is probably related to development. But I'm going to come back to this in a minute, because we actually have a lot to get through here, so I got to speed up a bit. But when we shut down these neurons without injury and we test baseline effects on somatosensation, there's no effect whatsoever. So I just wanted to point that out. So then what we did is, one of the things that we wanted to do, so we know nothing about the spinal cord mechanical allodynia circuitry in terms of the neurons that are involved at the time we were doing this, other than some PKC gamma neurons that had been identified by, the Basbaum group in the 1997, lamina 1, NK1 neurons. And then there was, a study that was done by Liu in Journal of Clinical Investigation, where they basically did paired recordings between the PKC gamma neurons to what they called transient central cells, to vertical cells, to lamina 1 neurons. So it was kind of sketchy, like what was out there, but it looked like a sort of getting a framework for what the mechanical allodynia circuitry would be. Anyway, we thought we would just take our own approach to try to look at what's in the mechanical allodynia circuitry by taking advantage of this excitatory dread and then looking at FOS expression. And so we activate just these neurons in the spinal cord, and then we looked at the FOS. So if we give the drug, the ligand CNO, that causes activation of the neurons, we don't actually see much happening over the baseline, which I'm not showing you here, controls. The way that we got, FOS activation is actually by then stimulating after the injury, stimulating after the CNO, or while it's, CNO's on board, stimulating the paw. So we're getting like because you remember the paw threshold drops, right? And so that's like allodynia or hypersensitivity. So anyway, we get this FOS activation, which we think is really reflecting FOS, the mechanical hypersensitivity that we've induced. And when we looked at what neurons these were, what we saw was there were a lot of neurons that had calretinin, and they were in interlaminate 2, where like IB4 or MRGD fibers go. And then we saw a lot of the PKC gamma neurons, which I said before, had been identified as being involved in mechanical allodynia by Alan Basbaum's group and others. And so then we wanted to know whether calretinin neurons could be in this mechanical allodynia circuitry. So we used the same approach, where we put in the dread, the excitatory dread, And then we just, asked whether we could induce mechanical hypersensitivity, and that's shown here. It does, and nothing happened with heat. So this is just this thin layer of cells right here above the PKC gamma layer. We're activating those, and it's causing, threshold drop. And so this is the framework that I was telling you about, where the PKC gamma neurons went to the transient central cells, whatever. Oh, no, sorry, this is a little different. Anyway, the vertical cells and the NK1 cells. And we put into the model now VGLUT3 cells in the deeper lamina 3 and these calretinin cells in inner lamina 2. And so this is kind of the framework that we were thinking, that it was this pathway. And then my postdoc wanted to do an experiment, which I thought wouldn't be that interesting, but, why not? I don't know. You wanted to activate the calretinin neurons and then look at FOS. I mean, it probably wasn't interesting to me because I didn't get it at first, what he was talking about. But then, after, we had some conversation, and then he said, okay, it became clear. I think it will become clear to you guys why it was actually, kind of a clever thing to do. That is, activate with the CNO, and you don't really see anything. I was thinking, we'll just see vertical cells and lamina 1 neurons. But it was that trick of how you need the mechanical input in order to see the FOS. And by needing the mechanical input, you're going back down from the, through the lamina 3, the deeper, and up again, to get this pathway. And so when we did that, what was surprising, because it would have validated, like, okay, then we would see the VGLUT3 neurons, we'd see the, PKC gamma neurons, the calretinin neurons, the vertical cells, and the lamina 1 neurons, but we didn't see that. What we saw instead was that there were a lot of calretinin neurons and really not very many PKC gamma neurons. And so then that got us thinking that the model that we had come up with, this sort of simplistic framework, if you're coming through and activating with low threshold fibers this pathway, you would expect to get these neurons. And so what we decided to do was look at different injury models, because we knew that the PKC gamma neurons were important for mechanical allodynia due to neuropathic pain, but it wasn't so clear that they were important in inflammatory pain. And when we did that, what we found, and this was published a few years ago, but basically what we found is that if we do a spared nerve injury model, we would see significant numbers of PKC gamma neurons expressing the FOS. So we do the model, and then we do the stimulation on the paw, and we get PKC gamma neurons and some calretinin neurons. And then when we do the inflammatory model, which is carrageenan in the hind paw, now we get more of the calretinin neurons and not so many of the PKC gamma neurons. And so that set up this idea that maybe, the pathway isn't a pathway, but it's a network, and it's differentially activated depending on the type of injury, inflammatory or neuropathic. And our hypothesis was that the calretinin neurons are important for inflammatory injury, and the PKC gamma neurons are important for neuropathic. And so we could test this because we, had this powerful inhibitory dread, and we could shut down the calretinin neurons. So here we're putting this inhibitory dread into the calretinin neurons in inner lamina 2, and then we're going to ask what happens when you shut down the neurons and you look at these different pain models. And so here we can, in a carrageenan model, CFA incision, we can get reversal of this hypersensitivity. Also mustard oil. Interestingly, we didn't see a reversal of the capsaicin-induced secondary mechanical allodynia. I don't know if you guys use all these pain models, but, anyway, I'll tell you, why this is interesting. And then we didn't get an effect with the spared nerve injury model. when we shut down these neurons. So this seemed to agree with this hypothesis that these neurons are important for inflammatory injuries, but they're not, but they're dispensable for a neuropathic injury. And then we don't see any changes when we just shut down the neurons, in baseline behaviors without injury. And then for the PKC gamma neurons, we wanted to test, are they required for neuropathic injury but not inflammatory injury? And here we ran into a little bit of a problem, which is that nobody, including us, seems to be able to make a reliable PKC gamma Cree line, although we may have one now. So we instead had to use this approach, which is to inject an inhibitor, a potent selective inhibitor of PKC gamma. And so this inhibition of PKC gamma, coupled with the FOSS experiments that we were doing, we're trying to say that implicates the neurons in an injury model. If we see reversal, so we see reversal with the spared nerve injury model, but we don't see it when we do any of the inflammatory models. And again, like I said, the FOSS doesn't activate the neurons. So we come to this conclusion, and I didn't show you, but also you can block capsaicin. So the capsaicin spared nerve injury, those two models can be, the allodynia can be reversed with the PKC gamma inhibitor, was shown by another lab, which is Reza Sharif Naini's lab in McGill. He also did that experiment. So, we come to this kind of idea that we have, depending on the type of injury you have, you're activating the network in a different way, and what's required to get mechanical allodynia or transmit mechanical allodynia in the dorsal horn is different. And so, I started out with just wondering why VGLUT3 knockout mice don't have mechanical hypersensitivity. And through this series of experiments, I'm now wondering, a lot of things about spinal circuitry and all of the, basically when I got into the field, to be honest, I was kind of overwhelmed with all the different pain models that people used and what was actually happening in these models and what people considered the models, are they chronic, are they not chronic, Anyway, it was overwhelming. And so one of the reasons why I kind of got attached to this idea, and honestly, my lab now says, no more models, we don't want it, there's too many models, we're testing all the models. But the goal, to me, is that, whatever's happening in the periphery when these injuries are occurring, is causing, a cascade of events that leads to the activation of the dorsal horn circuitry. And, there's seems to be somewhat characteristic, although there's probably a lot of variability, but characteristic or sort of signature events, for an inflammatory injury or a neuropathic injury. We need to do more different types of neuropathic injuries. But anyway, at the end of my talk, I want to talk about that a little bit more. But now, every time I talk to someone and they say they have a pain model or an injury model, I don't do the trigeminal system so much, but, a dry eye model or something, I'm or, the neuritis model that Jeff was talking about. It's what's happening when you have that model in the periphery? What afferents are getting activated, what kind of if there's an inflammatory response or other molecules that are involved, and then how is that activating the spinal network? And the reason I care is because, my original, what I thought was happening was that there was just a pathway, and no matter what you did, and you measured this behavior or this behavior, it was, basically a pathway. But I don't see it that way at all anymore, and I think it has really important implications for, therapeutics, because it's really saying that, at all different levels of the nervous system, different injuries are causing the activation of different circuits, which then you wouldn't expect necessarily. You would hope to find a place where you could intervene, and it would kind of go across these different types of injury, but it really needs to be tailored. So then I just want to wrap up a few, I have two more little stories. So basically then we decided to make this model instead, which is, showing the VGLUT3 neurons deeper down in the dorsal horn. And this idea that sometimes this goes really slow, but basically that, there's maybe a set of neurons that get activated if you have a neuropathic injury through some forms of, disinhibition of the circuit that are unique to neuropathic injury or not unique. And then you have a different set of neurons that are involved in the transmission of the allodynia if you have an inflammatory injury. And, I think there's some mechanisms here that are going on that are making these neurons hypersensitive, and it's one of the areas that I'd like to go into. Is basically, underlies how this part gets activated. Doesn't necessarily involve disinhibition, although there's probably disinhibition of the circuit in some capacity. But, for example, an A current, people had characterized like Rob Giroux, when you have an inflammatory injury, you get the loss of this potassium channel A current, and it converts the neurons. When you inject them with current, like in a slice recording, they're normally, almost all the calretinin neurons are delayed firing. And the prediction would be that it would then convert them to being tonic firing. And if they're tonic firing, they're more easily activated by input. And so it could, hyperexcitable. And so he demonstrated that in inflammatory injuries, this could be a mechanism for hyperexcitability of the spinal cord network. And these neurons, calretinin neurons, express that channel. So that's, for example, a hypothesis for the activation of the dorsal horn network in the setting of inflammatory injury. So so because of the developmental expression of VGLUT3, I didn't think we could study what those neurons did. So I wanted to find an adult marker. Of course, we did end up figuring out what those neurons do anyway, but we found this other population just looking at the Allen brain or spinal cord atlas. So I don't know if you guys are familiar with that, but it's a resource that you can go to and get a lot of information, although it's high throughput, so you have to take it with some skepticism because they're not in it for due diligence per se, as much as I think they care that it's accurate, but they're doing it in a kind of high throughput way. But anyway, I saw this pattern, and it was CCK, and it looked a lot like what VGLUT3 looks like if you do an in situ at the peak expression. And so we got these mice for CCK, and I should say that they're all, the CCK neurons are excitatory interneurons, and they're mainly in lamina 3, 4. And so we decided to study those and see what they do. And they have, when you activate them with the excitatory dread, it causes punctate allodynia, it causes dynamic allodynia, and it also caused heat hypersensitivity. So they seem to be involved in all three of these forms of persistent pain. And then we used our inhibitory dread, and we shut down the neurons, and we can reverse both inflammatory and neuropathic, both static and dynamic, and heat hypersensitivity. So these neurons seem to be important for, heat hypersensitivity and static and dynamic allodynia, whether it's inflammatory or neuropathic. So they're my favorite, neurons, basically. And so, and then when we shut them down, we don't get any acute phenotype, like change in baseline. And it turns out, and I'm not showing you here, but the VGLUT3 population is a subpopulation of the CCK population. And I showed you that the VGLUT3 population only seems to be important for dynamic allodynia. I think, basically that, I think basically, and maybe this is only, I care about this because I'm involved in these populations of spinal cord neurons, but I think we basically identified, a subpopulation of the CCK that's doing the dynamic, and not static, and there's another population that's doing the static, and there's another population maybe that's doing, that's doing the heat. Like, it could be segregated that way. And recently, you guys probably know that a couple labs have published the single-cell transcriptome of the dorsal horn. So one of the labs is, well, it's three. Maybe you could tell me if there's more studies that I missed, but one of them is at NIH, Ariel Levine's lab. And then another one is Patrick Enfor's lab in Sweden. And then Rubong Ji has a really great MD PhD student in his lab who did the somatostatin population of excitatory neurons in the dorsal horn. But anyway, it turned out that from this single cell transcriptome, and particularly we were focused on Patrick Enfor's because we were kind of collaborating with him, but that the CCK population is one of his branch points where, the way that they do the study is they basically, take single cells, a bunch of single cells, they sequence them, and then they try to group them based on the similarity of gene expression. And so for the CCK population, there are 3 main subpopulations. And so now we can go and try to isolate, each of these populations, subpopulations, and test what they do and see if they're functionally distinct. So that's something that we're looking into. But, so that gave rise to this model now where we have the VGLUT3, the CCK, and then we have a heat hypersensitivity pathway and the 2 pathways. And then, we're thinking about this, whether we can find heat hypersensitivity pathway using the same kind of FOSS, paradigm that I showed you before. I mean, my thoughts on the heat hypersensitivity, and you guys would probably know more than I do, but is, that it was just, primary afferents that are heat sensitive become sensitized and then sensitized somewhere near 2 outer 1, and there's not really a pathway that's, like a FOSS pathway. But the fact that the CCK neurons are in the deeper part of the dorsal horn, like 3-4, and they shut down heat hypersensitivity, but they're dispensable for normal heat sensitivity. It made me think, I'm still trying to figure out, how are these neurons involved in heat hypersensitivity? Like, how are they required if the pathway is just going into the, 2 outer and up? I guess it also goes down to 5, but we have an idea.
Well, I'm a little bit curious because I don't know that lamina 3 cells really respond to nudges.
Yeah, that's the other thing is that, when we did do some preliminary FOSS, we didn't really get that. But, FOSS, you can't take a negative of FOSS, to mean a lot. So that's, it's pretty quiet.
A lot of LTMs, so low threshold mechanoreceptor responses, but not really firm. Yeah, it's weird.
The CCK neurons do have like these long processes that go up to 1 in this like kind of odd way that I was trying to figure out what those neurons could be, because I was thinking that maybe there's some kind of just local, release of, but you'd still maybe expect to see, activity of the cell body in that scenario. But I was wondering if, somehow in an injury model, now you're getting exaggerated activation of these afferents going to 1 and they're releasing something, and that's, a requirement to get the activation. But it's still a mystery how these neurons are important. Okay, so now I'm going to wrap up with the last story, which is that, after looking at the calretinin neurons, the VGLUT3 neurons, CCK neurons, what else, PKC gamma neurons, we felt pretty confident, like we could, shut down mechanical allodynia, whatever model, right, in inflammatory or neuropathic with one of these populations. But I had gotten some money from the Diabetes Association because I wanted to make I wanted to use pain models that might be more clinical, I mean, this is what I thought at the time. Of course, now people would say, oh, that's streptozin. I think I got my review of a grant back, and they're like, that's nothing. That's not a model or something. But we also use genetic models. Anyway, there's a lot of robust debate in the pain field, I've learned. But anyway, I wanted to do something more clinical. So the American Diabetes Association gave me some money to look at mechanical allodynia that happens in that condition. And so when we did these different models of diabetic neuropathy and with the knockout mice, the VGLUT3 knockout mice, and this is the spinal cord only deletion, but we also did it in the regular knockout, we actually didn't block or affect whatsoever the mechanical allodynia. And so then I was thinking, okay, well, that's, not what I would have thought would happen. And so, this is like a polyneuropathy, I guess one could say. And so then, I'm not going to talk about that. So then we decided, well, let's look at some of these other populations that we've been manipulating and see what happens when we do those, use the model with those. And again, shutting down the CCK neurons didn't do anything to the hypersensitivity from STZ. Calretinin neurons, we didn't see an effect. The PKC gamma inhibitor, we didn't see an effect. We didn't bother to do like combinations of these or put them all together because it seemed like there wasn't any effect with, like at all for any of them. So but it's something that, a reviewer had brought up, I should say. But anyway, basically what this is saying to me is that this polyneuropathy, and the same thing happens with a chemotherapeutic model that we use, oxaliplatin, is that that's even different, than in the inflammatory and the neuropathic, the polyneuropathic, in terms of activating the spinal cord. And so then Sean, who's been in my lab for a number of years and does all the mouse behavior, came across this paper because he was interested in the diabetic neuropathy and diabetes. And it was a paper that was published a few years ago, and it was claiming that methylglyoxal, which is a byproduct of glycolysis, that it's responsible for the mechanical allodynia and heat hypersensitivity, or, the pain in diabetic neuropathy. And the way that they came up with it is that they looked at patients who had diabetic neuropathy with pain and without pain, and what they found in the plasma was high levels of this methylglyoxal in the ones that had pain. And then they did some mouse work, and they came to this conclusion that what likely is happening is that this methylglyoxal, which modifies ion channels like sodium channels or TRP channel, that it's causing sensitization of primary afferents, and then that's leading to central sensitization. And so, then Sean was, wanting to, test whether, what happens with methylglyoxal in our system, because if it's the cause of, if it's sufficient to cause the diabetic neuropathy, mechanical allodynia, we should be able to we shouldn't be able to block it, right? But what happened was we could easily block the pain induced by methylglyoxal. And so this is methylglyoxal, and then we use this GLO1 inhibitor. So GLO1 is the enzyme that degrades methylglyoxal. And it's thought that the levels go down in diabetics, and that's what's contributing to the raised levels of methylglyoxal. But we could block it easily, even this, which you do 2 weeks, and it lasts for, we considered it a little bit more, physiological than just injecting methylglyoxal. But we could, so that was, and then we tested with the CCK neurons, and we could reverse the allodynia induced by methylglyoxal with the calretinin neurons, and then with the PKC gamma, there was no effect. So this signature to us is pointing to an inflammatory type injury. So, and with methylglyoxal activating TRPA1, it's to us it fits. I mean, we haven't looked for, whether methylglyoxal actually causes, an inflammation, but to me, what this means is that the methylglyoxal is inducing mechanical allodynia like an inflammatory injury would, and it's not, sufficient for what's happening in diabetic neuropathy patients. And so for people that are looking to fix pain, or at least the allodynia, any allodynia. An allodynia that happens in a diabetic patient, shouldn't be just studying methylglycoxal, would be my take-home message. But what we did find was that the CCK neurons block heat hypersensitivity. I showed you that. And they do so with methylglycoxal-induced heat hypersensitivity, and they also do in the diabetic neuropathy model. And so my thoughts here are, when I found the knockout phenotype, I was working with Alan Bassbaum at UCSF, but I wasn't in his lab. I was in a different lab studying transporters, and then we just collaborated with Alan because, he knows pain. And anyway, he would tell me, like, forget heat hypersensitivity. Nobody, no, that's not a medical, need, or, it's not worth studying, kind of thing. I don't know if I should be saying this. Sorry, Alan, but anyway. So that was my impression, was like, okay, is that true? I don't know. How we all have to come up to speed on these things. But, what I was thinking about, more recently was just, because of this project, was that, heat hypersensitivity, like in these diabetic models, when people are complaining about, burning hands or feet sensation, it makes me wonder, because when we're looking at this heat hypersensitivity, in some experiments I didn't show you here, it's really like a heat allodynia. Like people say heat hyperalgesia, but it's evoked pain, or what seems to be pain, at a threshold that normally doesn't cause that response. And so then that makes me think, well, is it possible that heat allodynia, is happening in these patients when they have this burning sensation, because it's just being activated by, body temperature in some kind of, flaring way. I don't know, but then it makes me think, maybe blocking neurons like CCK neurons could actually, treat the burning sensation.
And that becomes almost a semantic debate whether detection, transduction at body temperature is allodynia or spontaneous.
Exactly. I had that conversation with Catherine Bushnell. It's evoked by body temperature.
In the TRPV1 neurons, right. You wouldn't be able to tell the difference mechanistically between ectopic activity and degenerating TRPV1 neurons, or sensitization of TRPV1 to body temperature. So I don't know if the postsynaptic, if the network in the dorsal horn would know the difference between those.
It depends on how it's wired. That's what we were trying to figure out.
If
I have no
it's the same
idea.
neuron that you're getting spontaneous activity from, then whether or not it's sensitization down to body temperature or ectopic activity is more of a biophysical epiphenomenon in terms, as far as the dorsal horn is concerned. You don't really care. The main issue is that your primary afferents are driving input into the dorsal horn, and then what happens in the circuits is what you're really worried. Does that make sense?
I guess so. Are you talking from a therapeutic perspective?
Yeah, I guess what I'm saying is you may not want to put too much emphasis on, especially in thermal, whether it's allodynia versus spontaneous pain. You're right that when, one of the characteristic features of diabetic neuropathy is that people would say their feet are burning and they can't sleep, but it's essentially spontaneous burning pain. But whether or not that's because you're sensing body temperature as painful, or whether you've altered sodium or potassium channels and you're getting ectopic activity, probably isn't really important for.
That sounds like a Steve Prescott
dorsal horn circuit.
comment.
Far be it from me to de-emphasize the primary afferent.
Is that resolved by putting it in cold
Good question.
Yeah, I've been told that it does have it can be dampened by cold.
Although that can reduce ectopic activity.
Yeah, I'm not sure it solves the problem. But I just think it's something that, people don't generally, what we've seen here looks like it could be, this heat allodynia theory could be activating these afferent pathways at temperatures that are normally not, in the absence of injury. And these neurons block that type of pain. And if that's what's happening in people who have, it might not be, it could be, ectopic activity. But if the same circuitry is activated in people that we're seeing here, maybe intervening in this way could be a treatment. No? We can talk more about that over dinner. Okay. I'm happy if people want to disagree with me. That's how you learn, right? You learn better what your arguments need to be, and then you learn what theirs are. But I just wanted to say one thing that's not, I have to go back to one slide and then we're over. It's not over because I have to show you the people that did this stuff. I want to show you. But what I just wanted to point out here with this, so the idea for me about this is that, like I said, that the inflammatory pathway, perhaps, and a neuropathic pathway, perhaps, and then this is my hypothesis for polyneuropathy. My hypothesis for the polyneuropathy, it could be because these vertical cells, they have long dendrites that go deep in lamina 3, and they get all kinds of input from C fibers, A deltas, A betas. That, if you were to just release the, inhibition on these vertical cells, then you could get activation of this, NK1, and you wouldn't need, all of this would be dispensable, right? It wouldn't matter. And so I'm wondering if the way that this network gets activated in a polyneuropathy, like the diabetic neuropathy, is that there's some mechanism of disinhibition or other mechanism which allows vertical cells to get activated directly. And then they don't need input from these guys. You can't block the pain that way. And anyway, that's my hypothesis. And so I'm trying to collaborate with somebody who's been looking at mechanism that shuts, or that, keeps the vertical cells down. And then the last slide, this is the most important one, is this one. Oh yeah. So Sean did all the behavior. He's been with me for a number of years. Sally is a Tsinghua University exchange student who came to my lab for 2 years. She's in the medical school there, and the medical school is like the number one or two in China. So these students are super smart and super motivated. Anyway, she came to my lab and worked with Cedric, who was a postdoc who led this project. And Cedric now has his own lab in France, which is, impressive because it's very competitive to get your own lab there. And then Jeremy was helping also in this project, and he's now a first-year student in CNUP program at the University of Pittsburgh, graduate program. And then these two just joined my lab and have been helping with just, some of the experiments to finish off the projects. But David studied pain in Brazil and with Alfonso Romero Sandoval. I don't know if you guys know him, but I think he's one of the chairs of the basic SIG APS something. Anyway, he's great. Alfonso's great and David's great. And then Priyo joined my lab from India, and he studied, he did in vivo recordings in primates in macaque with somatosensory defects, so looking at plasticity. And so he's going to help me take some of the stuff that we've been doing, and a grad student who joined my lab last year, Cynthia. The three of us, Cynthia, we're going to try to do some of the work that I've been showing you in mice, in primates. So that's what we're getting ready to do. And that's my talk. Thank you very much. Okay, anyone have easier questions than Derek asked?
So I might might have missed it if you kind of implied the answer or directly gave the answer. Do you think phenotype we're observing from the EU knockouts might be due to improper migration of a cell population in the spinal cord?
We did do some like kind of rough, stainings, but we didn't really count or anything to see if, any of the populations were missing from the dorsal horn. But I think, based on what VGLUT3, it seems most likely to me that there's some kind of, disruption in just, local connectivity is what I'm guessing happened in the knockout. But it's something that I'm interested in, and I've talked about it with Mark Bechet to do that because he studies spinal cord development. But I just haven't had the resources to do it. But I think it would be an interesting thing to study, for sure. But yeah, I'm guessing it's just synapses somehow didn't develop the connectivity properly. Did you want to ask, or you wanted to ask?
I have a question. I think at the outset of your talk, you mentioned something about VGLUT3, and there are two populations of humans, those that care about it. And I put myself in the camp of those that don't, but I got a little bit intrigued after you said that. So I'm just curious about what's the selectivity for VGLUT3? Is it selective only for glutamate, aspartate, sneak-in, glycine? And can that, whatever, regardless of what the selectivity might be, can that change as a function of entry and affect that?
I haven't really thought about that before. Most of the uptake studies with the V-glutes show that they're selective for glutamate and they don't transport aspartate or other. There's some, idea that they may transport phosphate now, and also they have a chloride. They're dependent on chloride binding for transport, and then there also may be a chloride flux. But yeah, I don't know. I didn't really think about changes like that.
I'm just waiting in part two of it. I'm curious what's going on in the other end, so many ends. What's happening in the skin? So the receptors like maybe a hormone versus mitochondria, some of these others that some may be selective to quantate, others may be selective to brush. Is that important? How might that relate to your work? Can that be predictive in some way? Like those populations show as opposed to how does that play?
Well, I feel like I'm kind of at the beginning of, trying to understand all of this myself, to be honest. So, those are the kinds of things that I would like to, consider. My first pass would be, if I had, money to, just do what I wanted, it would be to, try to look at that, in different ways, the afferents that are recruited in these different injury models and see, if there's some way, and then, connecting that with the spinal cord. My grant that I just got is to look at the connectivity within these populations and other populations of the spinal cord, and with primary afferents and with descending inputs, so we could get a better idea of just the general foundation of the connections in both normal condition and then changes with injury. So that's, going to take a long time to do. But, it's really about the nature of the injury, at, you're saying, these other factors that are also contributing, right, to the selective recruitment of the primary afferents and then the selective recruitment of the spinal circuitry. So I don't know. It's like a... But every time someone says something about I have this model, and then we see these changes, I'm like, I want to hear more. And then I'm thinking how could that be affecting, if these are, if these are injuries that cause allodynia, how would they be feeding? Can I predict? Because, what I wanted to say about the capsaicin, if you don't mind, is that, originally in the paper from Reza, who's a friend of mine, he used it as an inflammatory model when the reviewers wanted to see what happens in his condition if he did an inflammatory model. He said, I'll do secondary mechanical, with capsaicin. And we see it, he inhibited it with the PKC gamma, right, inhibitor. We inhibit that way, but nothing happens with the cowretinid neurons. And And so then I was looking through the literature because I'm like, and I was asking everybody, do you think capsaicin is inflammatory? And, I think what it comes down to is it depends, so under our conditions, according to Kathleen Saluka, and we need to do the experiments ourselves, but she had a paper where she injected subcutaneous and she didn't get, you get a little bit of a flare, but you don't really get the edema. And then she didn't see the infiltration of the neutrophils. She didn't see classic inflammatory markers when she inject capsaicin at the dose like similar to ours under the skin. But if you inject it in the muscle, and it also lasts a very short period of time, this hypersensitivity, it's only like for a few hours, couple hours. But if she injected in the muscle, then she gets a really long-lasting allodynia, and she gets inflammation. So the inflammation comes, but it comes after the allodynia is already gone when you do under the skin. So basically, this may be I may be overreaching here, but I almost feel like we can take our populations and, do like an algorithm that we could say, give me a pain model and I'll tell you what kind of injury it is. Because we've looked at things like, the one that everyone uses, CCI, right? What happens in CCI? What kind of model is that? Is that a model of inflammatory injury, neuropathic injury? I mean, in our system, CCI is even a little bit different. It's inflammatory, and it's like kind of polyneuropathic. Neuropathic slash. So, and in some ways you could imagine that probably makes sense because you're like, tying these things around, the nerve, right? And then you're getting probably what's happening. That's what we need to know.
And it only works with CAC-CAF, right? Not with synthetic.
We used silk sutures or something.
For the
Yeah.
Question, you're really focused on the A-beta mediated allodynia here, it seems. Right, and I'm curious in terms of what role you think the C fiber, injury of C fibers itself plays in allodynia.
Well, I'm gonna have to punt that one because I think it's actually a lot of fibers are playing a role. I mean, it's just simple to say that it's A betas because that's people can relate to the low threshold fibers. But there's low threshold A deltas that have been implicated. Those go to the PKC gamma neurons and
Or a reduction in threshold from the injury.
well, there is I think it was Jeff was talking about the mechanoreceptor in nociceptors, right? So there was if you read in the literature, is it low threshold afferents get recruited into the pathway for mechanical allodynia, or is it, higher thresholds drop their threshold, right? And so there's a paper now, I think it's on bioRxiv, so that this guy could preserve his pecking order or whatever, his claim. But it's my friend, his paper, but Rez's. But he's basically saying that he thinks that these normally the nociceptors are dropping their threshold.
Well, you can test that.
Yeah, you can test that.
You can block C fiber transmission
Right.
and see if the allodynia goes away. We've done that.
Yeah, and it's different for different models.
Right. So you're
That's the complicated thing. So in some models, you do a constriction, and it does seem to be, the A betas, assuming, the A betas are the only things that are, it's A fibers, right? You don't know. It could be A deltas. I mean, I think it's unclear, basically. It depends on the model. It depends on the type of whether it's dynamic or static, right?
or where you're giving your stimulus relative to where the injury is.
Right.
It could be an area of secondary hyperalgesia that's A-beta mediated, but if you go to the site of injury, it may be C fiber.
Right. Well, yeah, I mean, see, this is the thing is, again, to be honest, is, I'm just, if I went, when I wrote my grant, if I went into all the different variables, my grant would be triaged for sure in every single turn. So I just try to like focus on one aspect, and then I just try to keep it simple. But there's so many, possibilities, and so I don't want to introduce them all. It just will get too confusing, I think, for people. But I try to keep it, some things constant, like we're looking at the same kind of static allodynia with these different populations, and we're trying to be consistent with the time frame that we're looking at after the injury, at the peak of the allodynia or hypersensitivity, how you want to look at it. But, it's really a branching off point. It's like, it's just laying a foundation, and then on top of that, you can start asking questions. Okay, now that you've, now what happens if you look, what are the different fibers that are coming in? Because even, dynamic and static, right? Those are probably two pathways with different afferent inputs, and yeah. So it was just an attempt to keep it kind of clean, or not clean, but, simple or something.
So I keep thinking about what it means for the for the animal to have all these different pathways that can be activated for different pain models and different pain states. And so I was listening to a podcast the other week, and they explained fairly convincingly that we didn't have a word for the colour blue. Did you hear about this? We didn't have a word for the colour blue until we could make the dye. Right, because the only blue that we saw was the sky, and that was just the sky, and that was it. So even though we were seeing it, we didn't have a name for it. So I was wondering whether you could potentially, just as a thought experiment, I'm not suggesting we do it, right, but as a thought experiment, maybe you could have a word for all these different kinds of pain. So eventually you'd be able to go, oh yeah, my toe feels a little bit like a laminar 3 heat sensitivity. I'm wondering, is it like that? We don't have the words because we can't relate the experience.
It's funny because I was recently thinking the same thing. Not in the humorous way that you did, but is the sensation really, the behavior that we measure looks the same, but is the sensation the same? And it's probably not, right? Because it is involving different circuitry.
So do you think you could put your leg in a black box? And then you could have a doctor give you a certain injury, and you'd be like, "Oh yeah, that's definitely a carrageenia from 3 days ago." And then you hit me with a hammer. You think you could?
Let's try it. Volunteers. Well, on that note, we should probably wrap up. Anyone who's totally fired up to continue this conversation can join us.